Hello everybody, so my name is Faisal Mushtaq. I'm from the University of Leeds in the UK, and this is part of a symposium on addressing the challenges of global brain health and diversity through neuroimaging. And I'm going to tell you about a project which has diversity at the core, as around sort of tracking adolescent neurodevelopment in the Born in Bradford longitudinal cohort using EEG technology. So to give you some context then, in terms of what is the Born in Bradford longitudinal cohort, it's about 13,500 children who have been followed from birth from a socioeconomically and culturally diverse UK city, one of the largest UK cities, and also the youngest city in the UK. And we have already, through this sort of longitudinal birth cohort study, a range of sort of information on these participants that we've been collecting, including cognitive and genetic information. And we also have connected, linked datasets, which include health, education, and social care record data that we're able to map on and track the developmental trajectory of our participants. Now, thanks to some recent funding, we are able to continue tracking this cohort through a very critical period of neurodevelopment, adolescence, and obviously the next 7 years, in fact we are 1 year into this already, we'll be continuing to collect information on the measures that we already have and include new additional information from these participants around their mental health and well-being, their the amount that they engage with social media, digital screen use, etc., and be able to link that on to our sort of existing sort of records. Now, one sort of critical development within this sort of project is that now we are expanding this cohort from that 13,818 participants through to including these participants and their classmates. So that includes a sort of sample of approximately 24,000 participants, and that makes it one of the world's largest studies of adolescent development. So I mentioned that this is a project that already has a wealth of data, and this is a slide that just gives you an overview of some of the different categories of information that we have on the participants and their parents, and what we'll be tracking across sort of what we've already been tracking to childhood and through to adolescence. Now, this is a presentation at OHBM, where I expect that sort of many of you are looking here and sort of thinking, okay, well, where is the neuro data? What MRI, what EEG, what data do you already have? And unfortunately, we don't have any neuro data. And that, I think, is a major oversight. I think you'll all agree that that is something that should be desperately addressed. And this is an incredible, unique opportunity for us to be able to get novel insights into the developmental process. So what we have with the Born in Bradford project so far is really an incredible opportunity for future scientists to develop a more complete understanding of adolescent brain development. And in the long term, then we can sort of create models that predict later onset and progression of neurological disease. But a critical part of the story is missing. So here is the proposed plan, and I'm going to talk you through some of the proof of concept work that we've been doing so far and sort of describe our plans for addressing this sort of oversight. So one of the major challenges when it comes to collecting data from a diverse and large group of individuals is the issue of scalability. And we have this here as a very concrete sort of problem in our cohort. So here is our 13,500 participants plus the additional participants who we're now tracking from the age of 13 and above. And being able to collect data from that volume of participants is a significant challenge. You just cannot bring in 24,000 participants into the laboratory and collect detailed measures on them and do that in repeated fashion, given it's a longitudinal sort of project. So what do we do? How do we possibly address that issue? Well, we know we can take that kind of approach. We can take the sort of laboratory-based approach with a small group of sort of participants and take a snapshot of that, but that doesn't give you the full picture. We can also go into schools and we can set up laboratories, so pop-up laboratories where we can collect larger volumes of data. That makes it, addresses one of the significant sort of barriers in terms of who we sort of collect brain data on. It's typically people who are able and willing to come into a laboratory. So we address that. We address that through creating pop-up laboratories in schools, typically sports halls. We convert them and we create these sort of stations in which participants can come and collect data. That's not a real recording right there, if anybody's worried about the quality of the data. I'll come on to that in a second. So the pop-up laboratory approach gives us a slightly bigger snapshot of the cohort. Some of the piloting that we've done gives us a rough estimate that we'll be able to collect about sort of 4,000-ish sort of participants in that cohort through that approach. But what we are really interested in, and what's really exciting for us, is this possibility of being able to sample almost all of our participants to get a truly representative sort of sample of sort of children and young people in the city of Bradford. So how on earth do we go about addressing that problem? Well, so the answer, we think, lies in the use of low-cost commercial EEG technologies. So here is one provider, and this is a disclosure that we are working in partnership with Emotiv. We haven't received any funding, but we are in sort of close contact as we go about developing this project. So Emotiv, one of the world's largest sort of suppliers of commercial EEG systems, and they've demonstrated in the last year or so a proof of concept of being able to acquire EEG data remotely, online, and they did a sort of a small scale sort of study demonstrating sort of alpha suppression in a resting state sort of EEG task. And they've also built up sort of quality control algorithms to sort of ensure that sort of EEG signal is of sufficient quality. So that got us thinking. When we read that paper, we said, okay, well, how could we take that kind of approach and sort of implement that in our cohort? It seems that sort of that might be one of the sort of solutions for us. So this is just a figure showing sort of canonical eyes open, eyes closed topographical sort of distribution of the effect. You can see that it's prior to the sort of maximal alpha power. So the notable thing about commercial EEG systems is that they are meant to be extremely easy to use and targeted at people who are not sort of research EEG sort of experts who have sort of years of experience of applying EEG on participants. So in theory, you can have a complete novice acquire relatively good EEG data. So here's a video just sort of showing you in the background, sort of if you're not familiar with these EEG systems, this is a sort of a video of somebody applying it, a young person applying this sort of EEG system. And this is their low density sort of EEG system, this is the œpop-up, for those of you interested. And you can see there's a self-application of the EEG headset. So it might be clear to you now then in terms of where we're going with this. So the question that we have then is, can we take low-cost commercial EEG systems into schools in the city of Bradford to be able to acquire data from all of our participants? So how might we do that then? So this is our proposal. So our approach then is to take our participants from a classroom setting, pair them up, and have one act as an experimenter, one as a participant, reverse the roles, and in that way we can collect data from large volumes of participants. So just to sort of illustrate this then, so we have a lab-based data collection method where we can collect large volumes of data per participant, but we're constrained by the number of participants, so the throughput is relatively limited. But we're pretty confident that that's feasible. Of course, we've been doing laboratory-based EEG for a very long time. We've also, in our sort of explorations of pop-up laboratories, shown that we can collect data from simultaneously from participants from a classroom, pulling them out of the classroom and recording EEG. And in that way, we can collect a reasonable amount of data. And that has also sort of shown decent sort of ear signal quality. The question and the focus of this presentation is really around this sort of novel approach of semi-supervised peer application, where we can collect huge number of participant data but of course it's constrained in some ways in terms of the number of channels that we'd be acquiring and the amount of data per participant. So if you're following what's being proposed here, then you might have some questions around the feasibility and the validity of this approach, and so do we. and so did we when we started developing this sort of plan, and we came out with a list of sort of various questions that relate to sort of our participants who are teenagers who in many cases, 99% of cases, have never come across EEG before. How are they going to be able to follow instructions and record good quality EEG data? Are those participants who've got their friends testing them, are they going to be able to follow the task instructions? And given the fact that we are recording data simultaneously from 15 participants at a time, are there these other environmental factors, noise, etc., and electrical interference when you're recording from? Are those going to be issues in the EEG signal? Then there are fundamental questions around sort of who we will be able to acquire sort of EEG data from. So what about participants who generally historically been precluded from sort of EEG experiments, so people with extremely thick and curly and braided hair, for example. And ultimately the questions around sort of come around to sort of quality control and whether the signal quality that we are able to acquire and the sample size is sufficient trade-off for our other sort of approaches that I presented just now. So we decided to go ahead and implement this approach and test out whether this is actually feasible and valid. So what we developed is a protocol. We listed out some of the timings here in terms of how a session is undertaken. Effectively, it replaces a double science session in Key Stage 3 science in the UK. So that's in secondary schools, and it's curriculum aligned. So in this process of teaching participants how to acquire EEG data, we align that with some of the principles that they're learning in their science classes. So in terms of questions around quality control processes to ensure that the data that we get are sufficiently useful, we have three different sort of approaches to quality control. One is the built-in sort of processes designed by Emotive around impedance checking. You can see that on the left-hand side there, and EEG quality, which is a proprietary sort of algorithm sort of score, which gives you an indication. It's relatively easy, user-friendly, which is exactly what we need in this type of scenario. The second is around being able to track electrode position. So we want to ensure that there's a minimal amount of sort of variance between participants and their application of these headsets. So we are recording that information as well using a 3D scanner. And then finally, what we have are experts in the loop. So we have trained sort of EEG research associates who are on hand to be able to monitor sort of setup. So each person, so we typically have 5 people in a room for 15 simultaneous recordings, and they take on 3 of those sort of stations and monitor those and make sure that everything is going as expected. And we don't start recording until our trained EEG researchers have given the green light on acquisition of data. So the million-dollar question then is, what do the data look like? Well, so here's a power spectral density plot for a few of our participants. So I have to say we're at the very early stages of this sort of process right now, and between this recording and the presentation at OHBM, I hope we'll have added substantially to our data set. But so far we have about 50 participants, and there's a subset of these participants who all show, what we're seeing here is sort of the difference between eyes open in the blue and eyes closed in the orange. And you see for most of our participants we get this classic sort of increase in alpha power. But it's not all sunshine and roses. Of course, we also have a proportion of our participants who don't show those sort of characteristics, and in fact show quite messy sort of data. And that's sort of something that we sort of expected. It's something that we're sort of going to need to sort of quantify, and again come back to that sort of trade-off question to see whether this is actually worthwhile as an approach. I just want to show you here in terms of averaging across our 52 participants, and you get very clear differences in alpha frequency band. And when we then compare and benchmark this against other data sets, so in this particular case the TD brain data set, you see that this is exactly as we would sort of expect. So the initial signs are extremely promising for this approach, but there are still many questions that we need to be able to sort of answer satisfactorily before we scale this up. And here's the percentage of data that we have to remove following a pretty minimal pre-processing sort of a step using auto-reject. We lose about a quarter of the data for participants using eyes open and less using in the eyes closed condition. So in some cases it's a very high and sort of a large amount that's not going to be tolerable. So we've got participants there, some of whom were 80-90% of the data is removed, but we've also got lots of participants for whom the data is perfectly fine. So we have less than sort of 10% in a very large sort of proportion of our data. So signs are sort of promising. We think that this is a trade-off worth making, given the fact that we're exploring sort of 4 minutes of data for each condition. We still have sort of a decent amount of data left from which we can analyze these participants. And what we're going to do over the coming months is refine and tighten that protocol to minimize sort of further sort of data loss. But that's one of the sort of reasons why we're so optimistic and excited about this sort of opportunity is that even with some of those sort of data sort of quality issues, we're not going to be able to get absolutely everybody with high quality sort of EEG, but we hope that we'll be able to get a majority of participants through. And in that process, we have an opportunity here. We can engage with adolescents, typically who are quite difficult to be able to sort of recruit to scientific sort of experiments, and particularly in terms of the diversity of a sort of sample that we have. We've got an opportunity to engage and excite the next generation, and that is a focus in the way that we're developing our sort of workshops, effectively teaching them about neuroscience and sort of drawing on principles from engineering and data science. And all in all, we have participants who are actively involved in the scientific process. And you think about the sort of contrast between that and sort of having participants turn up to the laboratory and then sort of follow some instructions and off they leave. This is a fundamentally different way of sort of acquiring neuroscience sort of data. So I'm just going to end with this last slide in which we asked participants for their sort of thoughts on the experience. So we found that generally they agreed that it was relatively easy to use, they had a better understanding of EEG as a result of this sort of workshop. And then we also left open a sort of box about sort of their excitement and where they think this kind of approach could be applied. And you'll see some of those sort of comments there around being able to apply it in lower middle income countries, range of ages and cultures, and measure brain activity in different sort of populations that are more diverse. So I think that's again sort of points to the fact that this is an approach that can scale and then address some of the issues that we have in sort of human neuroimaging in terms of the lack of diversity in our samples. And so the last thing for me to just point out is that the majority of this work, almost all of it, has been done by Leila Kouara, a PhD in our group. And I'll just end by thanking our funders for their support for the group's research activities. And we are in the process of recruiting. So if you're interested in hearing more about this work, getting involved in this sort of work, then you can get in touch with me via email. Thank you for listening.